Welcome! I'm so glad you're joining me for this explainer. Today, we're diving into Chapter 9, all about stem cell culture and organ culture. We are literally talking about the basic building blocks of life and the kind of regenerative medicine that honestly sounds like straight-up science fiction, but it's happening right now in labs all over the world. So if you're ready to explore how these microscopic processes are totally reshaping the future of human health, let's get right into it.
Okay, let's dive into this outline. We'll start by meeting the stem cell, tracing a timeline of major breakthroughs, and classifying the different types. Then, we'll look at how we're actually curing diseases with them, move beyond single cells into organ culture, and finally, wrap up with the 3D future of medicine. All right, Section 1. Meet the stem cell, the building blocks of life.
Let's start right at the foundation, right? Before we can build these massive, complex organs, we have to understand the basic cellular bricks we're using. A stem cell is basically an immature or unspecialized cell. Think of it like a biological blank slate. And they've got two main superpowers. First up is self-renewal. They can just divide and make perfect copies of themselves to survive adverse conditions for really long periods. Second is differentiation. Basically, they haven't decided what they want to be when they grow up yet, so they can transform into a huge range of highly specialized cells: skin, liver, muscle, you name it. They're absolutely the body's ultimate wildcards.
But it's a profound thought. How exactly does a single blank slate cell, with almost no specific features at all, somehow know how to become the beating tissue of a heart, or the crazy complex neural network of a brain? This whole process of differentiation is exactly what scientists have been racing to figure out and control. Which brings us to Section 2, a timeline of breakthroughs, tracing the discoveries. And this brilliantly illustrates just how fast we're moving here. Fun fact from the chapter, the very first stem cells were actually isolated from blood cells. But just look at this explosion of progress since then. Back in 1998, we got the derivation of human embryonic stem cells. Jump to 2012, bam, a Nobel Prize for figuring out how to reprogram mature cells back into a pluripotent state. The very next year, in 2013, the revolutionary CRISPR gene editing tool was applied to them. By 2015, we're seeing actual clinical trials, and by 2017, scientists are generating literal organoids, like mini organs. The acceleration is just breathtaking. Moving to Section 3, types of stem cells, classifying potency and source. So, now that we know the history, we need to know that not all stem cells are created equal. We categorize them first by potency, which is really just a fancy word for ability. At the very top, you've got totipotent cells. Toti meaning total. These guys can differentiate into literally every single cell type in an organism. One step down are pluripotent cells, still incredibly powerful. They can become almost any cell type, except for those extra embryonic support tissues like the placenta. And finally, we have multipotent cells. Their abilities are a bit more dialed into a specific, closely related family of cells. For example, neural stem cells are multipotent. They're only going to give rise to nervous system cells, like neurons or astrocytes. We also classify them by their source, embryonic versus adult stem cells. Embryonic stem cells, as you might guess, are found really early on in the inner cell mass of the blastocyst. But here's the thing, actually scratch that, the name adult stem cell is kind of misleading. They're also called somatic stem cells, and they don't just come from adults. You can find them in kids or even umbilical cords. You have them all over your body right now, with a ton of them hanging out in your bone marrow. Their main day job is maintaining bodily homeostasis, keeping things perfectly balanced by preserving and repairing the exact tissue they live in. Okay, Section 4, curing with stem cells, real-world applications. So why does all this microscopic biology actually matter to you and me? Well, the applications are completely life-changing. By developing new cells in vitro, doctors are aiming to physically replace damaged tissues in patients. We're talking massive implications for treating things that currently have zero effective therapies: Parkinson's, type 1 diabetes, severe neurological disorders. Plus, studying these cells lets us uncover the actual root causes of genetic defects. And, this is absolutely crucial, we can test new pharmaceutical drugs on these cells safely, which drastically reduces our reliance on animal models. Let's move to a real-world cardiovascular scenario and see how this builds. Step 1. A patient suffers ischemia. That's a severe restriction of blood flow, resulting in heart muscle injury. Step 2. Under the traditional medical playbook, treatments are fairly limited. Surgeons can repair a blocked artery, or doctors can prescribe meds for fluid retention, but the heart muscle itself, it basically stays damaged. Now, Step 3. This is where stem cells totally change the game. Stem cell therapy doesn't just manage those symptoms. It actively aims to reestablish and regenerate the lost heart tissue and those blood vessels. It is a complete paradigm shift from just treating a disease to actually healing it. This shift is so important that the chapter specifically highlights this quote. In pharmaceutical industries, the stem cells have evolved as a suitable model to investigate the potential or mode of action of drugs and have become an improved alternative against the limitation of the ethical challenges of animal models. Think about that for a second. By using actual heart cells or neuronal cells derived straight from human stem cells, our drug screening gets way more accurate for human diseases, while completely sidestepping some major ethical dilemmas at the same time. Up next, Section 5, Beyond Cells, Organ Culture. Now, what's really interesting about this transition is the jump from the micro level to the macro level of entire tissue structures. The contrast between 2D and 3D culture is wild. Conventional 2D cell culture is totally fine for keeping isolated cells alive in a nutrient broth, but it completely lacks that complex in vivo structure. It's kind of like keeping individual bricks laid out flat on your driveway. 3D organ culture, though, that preserves the actual anatomical relationships and physiological functions. You're taking those bricks and building a functioning 3D city, complete with the whole microenvironmental infrastructure those cells need to interact properly. So the formal definition of organ culture is the development of a part of an organ or the whole organ itself using tissue culture techniques in vitro on an artificial medium. It mimics the parent tissue beautifully, but, and this is a huge but from the text, doing this successfully takes insane precision. Explanted tissues have to be handled with extreme care and literally rushed to the lab, ideally within minutes of collection, or the whole thing just starts to deteriorate. And that brings us to Section 6. The future is 3D, mimicking the microenvironment. All this rigorous, high-stakes science paves the way for the most futuristic stuff in the chapter. Researchers aren't just growing flat layers of cells anymore. We are seeing completely modern, next-level culture approaches. Things like multicellular spheroids, complex scaffold hydrogels, literal organs on chips that mimic human physiology. And yes, absolutely, 3D bioprinting, where living cells are literally used as the ink to print complex biological structures. So the crucial point here is that all this tech culminates in what we call organoids. These are self-organized 3D tissue cultures derived directly from those stem cells we talked about earlier, combined with growth factors and collagen. And the chapter gives this absolutely mind-blowing example. Scientists have actually surgically implanted an artificial liver organoid into the peritoneal cavity of a rat. No way, right? The ultimate holy grail goal here is that these organoids will one day have the full potential to replace diseased or failing organs in living human patients. Which leads me right to this final provocative thought. We started our journey today with a single, unspecialized, blank slate cell. And now, we're talking about engineering functional 3D tissues. With organoids and 3D bioprinting actively making the leap from theory into reality, how close are we to a world where literally no patient ever has to wait on an organ transplant list again? It's a staggering possibility for sure, and it's all built on the rigorous, beautiful science of stem cell and organ culture. Thank you so much for joining me for this explainer of Chapter 9. Keep learning, keep questioning, and I'll catch you next time.